Hello everyone, today we will learn about employing beneficial nematodes to prevent mature grubs of Japanese beetles from overwintering and stop the emergence of their next generation adults during next July. Adults of Japanese beetles are metallic green in color and their grubs are C-shaped and whitish creamy in color. Both adults and grubs of Japanese beetles are the most damaging pest of many turf grasses, vegetables, row crops, ornamentals and fruits. Adult beetles mostly feed on foliage, flowers and ripe fruits, and their grubs feed on the roots of turf grass and other host plants. The life cycle of Japanese beetles consists of 4 developmental stages, including adults, eggs, grubs, and pupae. The life cycle of Japanese beetle begins when adults emerge from pupae during July and August months. Emerged adults feed on the foliage, flowers, and ripe fruits, while feeding adults mate, and mated females then burrow into soil and lay eggs near roots of host plants, especially turfgrass. Eggs hatch into small grubs within a week. Hatched grubs immediately start feeding on the roots and develop into first, second, and third instars from August through September. However, these third instar mature grubs reduce feeding activity when they sense that temperature is cooling down and needs to move deep into the soil for overwintering. This happens when temperature reduces to 60 degree Fahrenheit or 16 degree centigrade in October and remains cold throughout the winter. These overwintering mature grubs start moving up in the root zones of their host plants and start feeding on the roots when temperature begins warming up in the March. These grubs continue feeding throughout the spring and then form pupae. Adults of Japanese beetle will start emerging from pupae in July and life cycle continues. So what are the precise timings for employing beneficial nematodes to interrupt the life cycle of Japanese beetles? Although 4 different life stages like adults, eggs, grubs and pupae are present in the life cycle of Japanese beetles, only grubs and pupae are susceptible to beneficial nematodes like Heterorhabditis, bacteriophora or Steinernema carpocapsae. Therefore, it is important to know the precise timing of occurrence of both grubs and pupae within the life cycle of Japanese beetles. As seen on this slide, the first, second and third instar grubs occur in August through September. Therefore, mid-September is the best time to apply beneficial nematodes to target and kill third instar grubs and prevent them from overwintering. Another best time to apply nematodes to target and kill overwintering grubs that are already moved back from their overwintering site into the root zone of their host plants like turf grass and started feeding on the roots and ready for pupation during April through May. This means if nematodes are applied in the turf grass in late April or early in the May, they will find and kill both grubs and pupae. As a result, the emergence of Japanese beetle adults will be prevented in July and the life cycle of Japanese beetle will be discontinued. What are beneficial entomopathogenic nematodes? Beneficial nematodes are thread-like, unsegmented, microscopic roundworms. They are also called beneficial nematodes because they are used for the biological control of insect pests of many crops grown in the greenhouses, fields, and gardens. They are also called entomopathogenic nematodes because the symbiotic bacteria carried by them are pathogenic to insect pests. There are two commercially available beneficial nematodes used for the effective control of Japanese beetle grubs include Heterorhabditis bacteriophora that carries symbiotic bacteria called Photorhabdus luminescens, and Steinernema carpocapsae nematodes that carry the bacteria called Heterorhabdus nematophilus. How beneficial nematodes can prevent Japanese beetle grubs from overwintering. After application, nematodes will move in the soil where they will find and mature grubs and enter into their body cavity through natural openings such as mouth, anus, breathing pores, also called spiracles, or intersegmental membranes. Once in the body cavity, Heterorhabditis bacteriophora nematodes will release symbiotic bacteria called Photorhabdus luminescens, or Steinernema carpocapsae will release Heterorhabdus nematophilus bacteria in the blood of grubs, where bacteria will multiply and cause septicemia that will kill grubs within 48 hours after infection, before the grubs will start moving deep into the soil for overwintering. And that's how beneficial nematodes will stop the overwintering of these grubs. The beneficial nematodes are used for biological control of Japanese beetles because they are active parasites of Japanese beetle grubs. After application, they will move quickly in the soil where they will find, infect and kill both grubs and pupae of Japanese beetles. Beneficial nematodes can reproduce inside the body of any insect larvae or grubs and recycle themselves. These nematodes are easy to apply. They are safe for handling, not harmful to plants, animals, and humans. Unlike pesticides, they do not leave any toxic residues on the produce, and therefore produce is safe to eat after its harvesting. Visit bugsforgrowers.com to gain practical knowledge about organic growing, insect pest control practice that does not harm plants, people, or pets. To read the blogs on biological control of insect pests of different crops, animals, and honeybee hives. Thank you for watching. Please comment, like, share, and subscribe. Look for next video on biological control of small hive beetles using beneficial Heterorhabditis indica nematodes.